This lesson is the fourth and final biomolecule of biochemistry. We have done carbohydrates, we have done lipids, we have done proteins. We are now going to do nucleic acids. Nucleic acids include DNA, RNA, and ATP. DNA, anybody know what DNA stands for, those actual letters? So close, close enough. Deoxyribonucleic acid, 2 words. Deoxyribonucleic acid. Maybe I may make that a bonus question on your test. Spell out what DNA stands for. Spelling counts. What does RNA stand for? Anyone? Come on, Will, have some guts. Ribonucleic acid. And you all remember this is what the mitochondria and the chloroplast make. What is ATP? Adenosine triphosphate. How many phosphates are there? How do you know? See? Using that Latin. Okay, these are polymers. We'll get to them in a moment. The one I really want to focus on this period is DNA. That's what I'm really going to talk about. I'll talk a little bit about RNA and ATP, but DNA is going to be the main attraction. DNA was discovered. Now, key word here, it was discovered by a woman named Rosalind Franklin. However, she discovered it. There's a couple guys named James Watson and Francis Crick. They used Rosalind Franklin's work, not necessarily with her permission, to figure out what the shape of DNA was. They made like this big 9 or 12-foot model of a DNA helix back in the 50s. And they won the Nobel Prize for it. They didn't win the Nobel Prize for discovering it. They won the Nobel Prize for learning its structure. So James Watson and Francis Crick, oops, spelled his name wrong, used her work to realize the shape of DNA. So they kind of ripped her off. It's one of the dirty stories of molecular biology. They won the Nobel Prize in 1953. So guys, DNA is still relatively new. Your grandparents were, when your grandparents were kids, it wasn't discovered yet. That's like how new it is. It's still pretty new. What I actually just read, there was a man in Tampa who was accused of murdering somebody in the 80s. And he's been in jail since the 80s. And they just released him because of DNA evidence. Like DNA is the end all be all of proof. You can't deny your DNA. When you want to learn what your ancestry is, like you send a DNA sample, you want to find out if your ancestry is African or German or Italian or Norwegian or Vietnamese. You send it to these companies and they can figure out just by reading your DNA what common traits you have. They don't ask for a picture of you. They don't ask for the shape of your nose. They don't ask for what your hairstyle is. They ask for your DNA because that tells everything about you. You ever heard of the TV show, The Maury Povich Show with this guy? Let me see if I can find it. Let's see, where is he at? There he is. Anybody seen that guy on TV? Yeah, it's the You Are the Father guy. That show's been on since I was in school. It's the Maury Show. I'd say by far the majority of his shows are all about paternity tests. There's a woman that comes on TV and she says, I need to find out who the father of my child is. And you got maybe 1, 2, 3, 50 guys. And they're like, that baby has your nose, that baby has your ears, that baby has your lips. The man, what's the guy always say? That baby don't look like me. And then they do the paternity test. He pulls out the folder. He's like, the results are in. And when it comes to 12-year-old Aiden, Reggie, you are the father. I told you that baby is yours. They're looking at the DNA tests, not the feet, not the fingerprints, not the way your teeth look. It's your DNA. And your DNA tells all about you. If you haven't watched that show, it's an experience. All right. So let's continue talking about some of nucleic acids. Let's talk about the elements found in nucleic acids. By the way, the Na in DNA and RNA is nucleic acids. The elements are carbon, hydrogen, oxygen, nitrogen, and what element have we not used yet so far this week? Phosphorus. Phosphorus is the only macromolecule that actually has, DNA is the only macromolecule that has phosphorus. So if you found some mystery molecule and it had CHON and P, done deal, it's nucleic acid. What if it had CHON and S? Right. If it had nitrogen, here's a very important question. My AP kids just took a test on this. If it had nitrogen, what macromolecules could you narrow it down to? Nope. Fat does not have nitrogen. Lipids are fat. Protein or carbohydrates don't have nitrogen either. You gotta remember that. Nitrogen, as in nitro, N, the letter N. Nucleic acids and proteins. Those are things you guys have to study. Test on Tuesday. All right. One sec. Sure, we'll do this. This protein. Okay. The monomers. Of nucleic acids. This is going to take about 10 minutes. It's not just as simple as saying what it is. They are called nucleotides. Morgan, how many parts are there to a nucleotide? You remember? One. Two. Three. Still remembers it. Good. There are 3 parts to a nucleotide. The first part is called the phosphate group. It is the same in DNA and RNA. It doesn't change. And it looks like this. A phosphate is PO4 with a -3 charge. If you were in chemistry, you had to memorize that polyatomic ion. It's PO4. That's a phosphate. The second part, everybody, is a pentose sugar. What number is represented by the prefix pent? 5, like the United States military's headquarters is the Pentagon. A pentose is a 5-carbon sugar. What type of sugar is glucose? What size is it? What shape is it? It's a hexagon, so what do you call that? Hexose. What does ose mean? Sugar. Now, this does differ from DNA to RNA. Guys, in the last few months, I've heard this mistake happen, and it's kind of concerning me that it's happening a lot. Na stands for nucleic acid. So when you hear DNA, you got D nucleic acid. Well, what is D? D stands for deoxyribose. This sugar is the pentose in DNA. And what do you think you call the sugar in RNA? Deoxyribose is DNA. What is RNA? Ribose. This is the pentose in RNA. Here's the mistake I've been hearing. I remember this as recently as April, right before my AP kids took their test. I would say, guys, what sugar is in DNA? And some kids would say, ribose. That's what the R is. That's RNA. DNA is deoxyribose. Careful about that. Apparently that's a mistake, a misconception. The pentose looks like this. And these carbons have numbers. 1, 2, 3, 4, 5. They're called primes. You'll learn this in unit 6. 1, 2, 3, 4, and 5 prime. Okay. And then the third and final part, a nitrogenous base filled with nitrogen. That's how they get their name. There are two classes of nitrogenous bases. First kind is called a purine. It is a 2-ringed molecule. It looks like this. It's like a hexagon and a pentagon. This includes the nitrogenous bases adenine, and guanine. You may remember that DNA is just 4 letters, A, T, C, G. This is the A and the G. They are called purines. And the other type is called a pyrimidine. A pyrimidine is a 1-ringed molecule. This includes cytosine and thymine. I want to tell you how I remember this, but I'm going to let you guys catch up. There's actually another one that's after thymine. I'll let you guys catch up for a moment. Yeah, that's what I'm going to get to. So if you didn't just hear Aiden, he said, isn't it the U one? Yeah, that's what I'm going to get to. So the letter Y is not that common of a letter. You don't see it in every word. It's kind of a rare letter. But you see Y in pyrimidine. You also see Y in cytosine and thymine. Perhaps I can help you remember them. Thymine and cytosine are pyrimidines. But as Aiden just was alluding to, in DNA, the letters are A, T, C, G. What are the letters in RNA? One of them is different. Anybody recall? No, RNA does not have T. So what are the 4 letters? A, G, C, U. It goes A to U, G to C. U stands for uracil. Uracil is also a pyrimidine. Yep. Put the door stop in the door and the entrance door. The door's locked. All right. If you were to draw all three of these parts together, here's how they would look. That is a nucleotide, everybody. You have your phosphate. You have your pentose. And you have your nitrogenous base. When they combine, they form one nucleotide. That is the monomer of nucleic acids. Now, if you wanted to simplify this, here's what I would allow. You have the P for phosphate, the S for pentose sugar, and the N for nitrogenous base. That is a nucleotide. Pretty much the same thing, just a little more simplified. Now, if you wanted to draw a bunch of them together to form a helix, you just have to have them repeat themselves. I see a lot of pens moving, so I'll wait. It's no problem. Okay. I want you guys to stop writing for a moment. I want you to just watch this. I'm going to show you how you actually make DNA or RNA. You just take all the nucleotides, you just add them repeat. So watch this. Just watch. Can anybody tell me how many nucleotides I have drawn? How many nucleotides do I have? How many, Natalia? 4. That's right. 1, 2, 3, 4. Very good. Now, that's something you'll just have to be able to show on your homework is how do these nucleotides bond together? They bond together mainly through this. I want you, this is going to be really important for not just this unit, but for future units. This part that I'm highlighting in yellow, this is what we call the backbone of DNA. It is repeating sequences of phosphate groups and pentose sugars. They just repeat themselves over and over. I'm going to walk around. I want to show you all this model of DNA I have here. I want you all to clue in on, I'm missing a little part. Nope. I want you guys to look at this model I have. These red parts are phosphates. Every little red part on the side is a phosphate group. Everybody, please look up here. Every one of these is a phosphate. All these black parts here, 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 those are sugars. I want you to notice how it just goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. These yellow bonds are covalent bonds. They're holding the phosphate to the sugars together. If I were to flatten this out in a two-dimensional shape, it kind of forms a ladder. If you've ever walked up a ladder before, you know that you put your feet on the steps, you put your hands on the sides. The sides, where I'm holding here, everybody look, these are the sides. This is what we call the backbone of DNA. It is made up of alternating sequences of sugar, phosphate, sugar, phosphate, sugar, phosphate, just like that. This is the backbone of DNA. They're held together by covalent bonds. And they are strong, meaning they shouldn't be broken. You don't want to break these bonds. Things like radiation, dangerous chemicals, they can hurt your DNA really, really bad. You don't want those bonds broken. That can lead to bad things like mutations and damaged DNA, stuff that can really ruin your day. Okay, we have about 7 minutes. These parts here, these are the bases. I'm going to hold up my model up here again. The middle part, let's say this is a ladder. Check this out, this is going to look really corny. I'm walking up a ladder. Fell down. Ready? Here we go. I'm going to crawl down the ladder. The parts that I'm pinching right now, these are the bases. If I was walking up a ladder, this is what I hold on to. This is the middle. You should be able to do this. Check this out. You want this to be able to happen. If I should be able to do this, should these bonds be strong or weak? They should be weak. You want to be able to open up DNA so you can copy it. If you can't open it up, you can't copy the DNA. Whenever you guys hurt yourselves, you damage yourselves, or your cells worn out, and you have to replace them. Dylan is going to go dirt biking today, and he's going to get some road rash and really skin up his leg. That is damaged muscle and damaged skin that needs to grow back. That means he needs to regrow some DNA. This is how you do it. You open it up and you copy it and make more. Some teachers have these books that they keep in their cabinets. And in the book, there's a bunch of worksheets. You can't make a copy of the worksheet if you can't open the book. You can't make a copy of DNA if you can't open the DNA. So it looks kind of like this. Here's the backbone of DNA. Here we have adenine. I'll make adenine for A, so we got A's. Put some T's in there, some G's, and I'll put in some C's. Do you guys know what pairs up with every single time you have an A? What? T. But we're going to do DNA, so we're going to have T. We have G, and then some C's. Then the other side of DNA right there. In DNA, A is always with T, C is always with G. In RNA, A is always with U, C is always with G. You want to be able to do this. Watch this. You want to be able to open it up so you can copy it and make more. The bonds that hold these together are weak. Bonds that hold bases together are weak. They need to be able to be broken. They're hydrogen bonds. You want them to break, at least with the right breaker, with the right enzyme. Okay, we have about 5 minutes, I think. Let you guys get caught up. We'll start to conclude. Okay, let's start to wrap things up. What are the monomers of nucleic acids called? Nucleotides. What are the polymers of nucleic acids called? We have DNA, RNA. One we didn't get to today, we'll have to do it later, is ATP. What is the purpose of nucleic acids? Well, depends on which one you're talking about. DNA stores genetic info. You are the father. Yeah, prove it. Your DNA is almost an exact match to this child. We have 5 guys here, 5 possible fathers. Your DNA matches this child's DNA way more than any of these other men. It matches it so much that you must be related to this child. You're the father. These other guys, their DNA doesn't come close. Yeah. I'm gonna have to go back to it at the end, Olivia. I can't go back for one student right now. We need to finish this up. RNA produces proteins. And ATP is cellular energy. Makes energy for cells. It's made by the mitochondria. What is the shape of DNA? What do you call that spinning staircase structure? It's a double helix. DNA is a double helix. Kind of looks like this. Its shape and structure allows it to be easily stored in cells and easily replicated. All right, and that is it. 1 second.